Good afternoon, everyone. My name is Lucie Wilson, and I'm a first-year PhD student at Sheffield Hallam University. I'm here today to talk about my PhD project, which is to investigate the impact of low-level carbon monoxide, or CO, on the human brain. And I'll give an overview of the techniques that I have been using in my research so far. I'd first like to say a huge thank you to the CO Research Trust for inviting me to speak today, and to all of you who've joined to listen. So as I've mentioned, I'm approaching the end of my first year in a 3.5-year programme. For this project, I'm particularly interested in studying lower levels of CO, sufficient to raise exhaled CO levels to between 3 and 6 parts per million, to see what impact this might have on healthy human volunteers. The project has been designed to include both TCD and MRI imaging techniques to explore how blood flow velocity and response may alter in the presence of CO. This combined approach is also a novel way for me to consider the impacts both from a vascular and neurological standpoint, which I'll delve into a bit more later. This talk will showcase some interesting findings we've made so far using both TCD and MRI methods. So why are we looking at lower levels? We understand that most individuals are exposed to ambient levels of CO every day, for example, due to the burning of fossil fuels or from transport emissions. Additional factors such as tobacco smoking also increases an individual's exposure to CO dramatically. The cardiovascular and central nervous systems are particularly vulnerable to CO-induced hypoxia due to their high oxygen requirements. Despite government strategies detailing an acceptable level of atmospheric CO to be around 6 to 9 parts per million, far lesser levels of CO have already been linked with disease development, such as heart disease and stroke. Although these levels might not be sufficient to induce hypoxia, researchers found that levels of only 3 parts per million were sufficient to cause changes in brain function. This is notably far below what is currently considered to be a normal or acceptable level, and this is the area that I was most keen to explore further with my PhD project. With the knowledge that low-level CO has been linked to cerebrovascular diseases, we started to look at why that might be. As mentioned, the central nervous system is particularly susceptible to hypoxia, and we know that acute high levels of CO will cause massive damage to an individual's central nervous system, and this is typically expressed in symptoms such as headache, nausea, and dizziness. We also know that a smoker will exhibit an increased baseline carboxyhemoglobin level in the blood, but does that result in an increased tolerance to CO compared to a non-smoker or not? We also don't yet know how the chronic low levels of CO that we are exposed to every day impacts our brain function, and can existing damage related to tobacco smoking be reversed to any extent if baseline carboxyhemoglobin levels are reduced? If levels of only 3 parts per million have been previously linked to altered brain activity, what might the long-term impact of this be for society? These are the questions that I want to try and answer with my PhD project. Today, I'm going to break down the 2 main techniques that I have been exploring as part of my study, and I'll also outline how they will be used in the future research. I'll begin with the functional magnetic resonance imaging, or fMRI, component of my study. I'd first like to give a very brief overview into what MRI is and how the functional component fits in. MRI is a non-invasive type of scan that uses a strong magnetic field and radio frequency pulses to visualize internal structures. Functional MRI, or fMRI, is especially focused on the brain and looks at changes in cerebral blood flow during certain activities as a marker for cognitive demand and neurological function. Essentially, an active part of the brain will require more oxygen and therefore more oxygenated blood. There is a specific technique in fMRI used to measure this, termed blood oxygen level dependent or bold fMRI. This technique relies on differences in blood flow between brain regions to identify areas of high and low activity. With an action, cerebral blood flow increases, and so we get a big influx of oxygenated blood to that area. This predictable pattern of blood flow response is called the hemodynamic response. As you can see from the image on the right side of the screen, blood flow usually peaks around 5 seconds after an event has occurred, and then it will tail off quite quickly. Bold imaging picks up on the influx of blood, as oxygenated blood has different magnetic properties to that of deoxygenated blood. This is what generates our bold MRI images. I'll now move on to some of the findings that we have made using this technique so far. Using software that is specifically designed for use with fMRI scans, we have completed the analysis of some raw fMRI data that relates to cognitive function. The human volunteers in this study were non-smokers and in good health, and they consented to receive both an air and low-level CO intervention in a random order. Participants were connected to a custom-made breathing circuit, and either air on its own or a small amount of CO mixed in air were added to the breathing tube to increase their exhaled CO levels. All participants received fMRI scans and were supported to perform a basic reaction time task both before and after the air or CO intervention was given. The reaction time task consisted of the participant pressing a button with their right hand as fast as they could when a flashing dot appeared on the screen. The bold scan technique was applied here alongside structural scans that were used for anatomy. The findings of this analysis were really interesting. Firstly, all participants showed a significant increase in expired CO after CO inhalation, which was to be expected, and this is visible in the first figure. Secondly, the reaction time averages were taken for all participants and grouped by the type of scan they had, as is visible in the second figure. All participants demonstrated slower reaction times in their second scans compared to the baseline, but the reaction times recorded after CO inhalation were slowest overall. Side by side, we can see that where exhaled CO levels were highest, reaction times were in fact at their slowest. These are mean analyses to show the activation for all conditions in the cerebellum region. The analysis that I completed here results in the generation of colour-rendered statistical maps that have been superimposed on a standard brain to be precise. You can read these as images or maps of neural activation. You can see that there is consistent activation across the cerebellum in all 4 scans, which makes sense as this part of the brain is responsible for movement execution and coordination, but also for sensory processing. So it was likely that the cerebellum was activated in response to the pressing of a button during the reaction time task. As you can see, there is a great deal of overlap between each condition in terms of which area of the cerebellum was activated. This is reassuring as it suggests that the participants performed the task in the same way. Furthermore, we can see that whilst the amount of activation seems to increase in the second air scan compared to the baseline air scan, there is actually an observable reduction in bold activation after the CO intervention compared to the baseline CO scan. You can see that this scan type has smaller activation areas or fewer coloured blobs, and also as the colour coding is leaning towards red, this indicates a lower Z-stat, or you can think of this as a less significant result. The level of activation visible is much smaller here than in any other scan type, suggesting that the presence of low-level CO influenced cerebral blood flow in some way. Another region that I found to be largely activated was the motor cortex, which is situated towards the top of the brain on both sides. We can see that the activation is localized to the left side of the brain, noting that these scan images are flipped. The motor cortex is responsible for movement, and so we would expect to see activation in the left-sided motor cortex as the participant was pushing a button with their right hand. Whilst this region was again activated consistently in all the scan types, we can see a big reduction in signal in the post-CO scan type compared to all of the others. To reiterate, we can see this because not only are the blobs smaller, they are also more red than yellow. This again suggests that the presence of CO impacted the cerebrovascular response in this region. The previous results showed average regions of activation for each condition individually, but we are interested in seeing if there are any significant differences between the protocols, for example, between the AIR and the CO scan days if we compare them. The red-yellow scale that is presented here indicates where cerebral activation was lower following CO compared to the activation that was visible following just AIR. This means that in this particular brain region, the CO caused the bold signal to significantly drop. Importantly, this was identified at a significance threshold of 3.1, which is regarded to be a very stringent threshold. We are confident that these findings are therefore robust. The area highlighted here is the middle temporal gyrus, which is located in the temporal lobe. Research has shown that the temporal lobe is often a main area for cerebral damage following CO toxicity, but here we see reduced blood flow even with a very low dose of CO. Temporal gyri are often involved with multisensory processing, like what we have seen before with the cerebellum. It is a region with lots of different functions, and the superior part of the temporal gyrus may be involved with object perception, either in regards to the dot flashes that were shown on screen or with the physical touching of the reaction time equipment. Lesions in this region result in impaired exploratory visual search abilities, and interestingly, impairments in the right side of the temporal gyrus have been linked to autism and also to the development of Alzheimer's disease. So to summarize, the participants in this study experienced a very low dose of CO that was sufficient to raise their expired CO levels by around 3 parts per million. Coupled with this, we also witnessed increased reaction times, so participants were on average slower after CO inhalation compared to all other scans. We saw a significant reduction in middle temporal gyrus activation, but also reduced cerebellum and motor cortex responses as well. Obviously, this is a really exciting find, and we are building a list of things that we want to explore going forward in our research based on this. The results of this data has informed the next steps of my PhD project. I will shortly be recruiting for participants for more fMRI scans, but these will take place at the new PET MRI facility at the University of Sheffield. Not only does this scanner have improved spatial resolution, but we're also increasing the complexity of the cognitive tests that are performed within the scanner, as the more complex the task, the more energy is required by the brain to complete it. I am planning to incorporate a specific type of cognitive test, which is called an NVAC, and this is essentially a memory-related task. Participants will still be asked to give their responses as quickly as possible, and so the test will have a reaction time component to them as well. In addition to this, I'm planning to complete different scan types, as well as the bold fMRI scans as previously discussed. This includes a method called arterial spin labeling, or ASL, which is a technique that we can use to quantify the amount of blood we see traveling to a particular brain region, and this will allow us to draw more conclusions from the data. Not only will I be able to suggest where the blood has traveled to, but I will also be able to find out how much blood actually traveled. Diffusion tensor imaging is another technique that I will be using in my future work, and this helps us to visualize the white matter fibers within the brain. This generates the really colourful types of images that you can see in the top right-hand corner of this slide. The fMRI results are really exciting, and it can show us a lot with excellent spatial resolution. However, we understand that its temporal resolution is not as good. In order to observe what the impact is of the CO on a more granular level, in other words, what is the instant effect on the vasculature after it's been inhaled, we required a technique with more enhanced temporal resolution capabilities. This brings us to the second aspect of my research, which involves the transcranial Doppler ultrasound technique, or TCD. The TCD scans, coupled with fMRI scans, will be completed on a large pool of participants, both smokers and non-smokers. The TCD is useful as it provides a real-time view of cerebral blood flow velocity. So in this way, I can see if blood flow speed increases or decreases in response to an intervention. Participants will again receive a very small amount of CO mixed in air to raise their exhaled CO levels by around 3 parts per million. We have designed it in this way to identify if there is a difference in baseline velocity measurements compared to post-intervention results. I have put up a picture of the TCD equipment that we are using on the screen so you can see how portable it is. We are using a small transducer probe, which is the black object on the right, and this is especially designed for use with transcranial Doppler scans, although you can buy other probes to scan different areas of the body. There are a number of areas on the skull that have a lower bone density, and so the ultrasound waves are able to penetrate through. But the temporal window is most commonly used, and this is labelled as number 1 on the skull on the slide. TCD can be used for any number of examinations, including in acute cerebral events, such as diagnosing ischemic stroke. And it provides an alternative yet complementary view to structural imaging. It is our aim to combine both the TCD and MRI techniques to get a novel perspective on both the vascular and neurological impact of low-level CO on the brain. So how exactly does TCD work? Well, as mentioned in the slide before, TCD requires a low-frequency probe that emits ultrasound waves. These waves that travel through the thinner windows in the skull are then reflected back from the moving red blood cells within the vessel at a different frequency. The software is then able to calculate blood flow velocity and spectral patterns are formed. I gained this spectrum by scanning the transtemporal window, which can be used to access a number of important vessels, including the internal carotid artery bifurcation, or the ICA bifurcation. This vessel is particularly interesting to look at because it actually branches off into 2 other major arteries, and so it shows bidirectional blood flow. In the center, we see our midline or 0 line. The spikes that are going above indicate flow towards the probe, and the spikes that are below the 0 line indicate that there's blood flowing away from the probe, so it's going in the opposite direction. The other way that we can tell the direction of the blood flow is by the colour-coded bar that I've put just below the spectrum. This tells us that where it is red, blood is flowing towards the probe, and where it's blue, blood is flowing away. The individual spikes that you can see are termed waveforms, and these essentially occur with every pulse of blood. When multiple waveforms are recorded, this is termed a spectrum or a group of spectra. The numbers on the left-hand axis tell us the distance that the pulse of blood has travelled in centimetres per second. So the higher the spike, the further the blood travelled in that particular pulse. This spectrum of the ICA bifurcation shows the baseline blood flow with the participant holding their breath in the middle, and this is where the waveforms become shorter. A shorter waveform tells me that blood velocity has reduced as the blood didn't travel as far as it did at baseline. Once the participant began breathing again normally, the waveforms increased in height, demonstrating that velocity returned to baseline levels. This was a great task to perform to evidence the sensitivity of the TCD equipment to changes in cerebrovascular resistance. The performance of a breath hold is a very common physiological challenge and is widely used as an indicator of vasoactive response. In a typical case, a breath hold of 8 to 10 seconds will cause an increase in CO2 in the blood as oxygen levels are reducing. This kickstarts a chain reaction that results in cerebral vessel dilation. This is the body's mechanism to increase cerebral blood flow to maximize oxygen getting to vital areas when oxygen is reduced for longer periods of time. In this instance, a short breath hold resulted in a normal vasomotor response, and I've highlighted with the box where exactly cerebral blood velocity was impacted almost instantly. As mentioned, this test evidences how sensitive the TCD equipment is for real-time monitoring of the vasculature and why it can be such a useful addition to our research. It is likely that volunteers will be required to perform a short breath hold as part of their baseline TCD monitoring within the study to ensure that vasoactive responses are typical and reliable between participants. We have also started running our pilot CO protocol, which consisted of using a 2-litre bag of CO mixed in air, sufficient to raise exhaled CO levels from 2 to 4 parts per million in this particular case. This is comparable to the levels used in previous fMRI data that I've discussed. The gas is delivered via a custom-made breathing tube and administered over several minutes. Here, I took repeated TCD measurements every minute after the initial exposure to see how blood velocity measurements were affected and how that changed with time. You can see that as expired CO levels increased, blood velocity reduced. This is evidenced by the waveform peaks reducing in size further away from the original baseline in the first spectrum. This is a small increase in expired CO, and to a level that we would consider to be relatively minor, but we're already observing a change in the cerebrovascular response. We cannot say for certain, but it is likely that as CO is a vasodilator, the presence of even a small amount has dilated the blood vessels, and velocity has therefore reduced. We are hoping to be able to match this data with the arterial spin labeling scan data that we will obtain with the fMRI scans, so that we can calculate exactly how the cerebrovascular bed is responding. So, what else have I been getting up to during my first year? With the baseline data I gained, I was fortunate enough to develop a poster for the launch of the new Sheffield Multimodal Imaging Centre, or SMIC, at Sheffield Hallam University, which I am displaying for you now. SMIC is a leading research and development facility at SHU that recognises innovative techniques such as the TCD. With this poster, I was able to talk a lot about the actual technique for TCD and how I was using it for my research. But I was also able to demonstrate how sensitive the TCD equipment is to the physiological tests, such as the breath hold. We are now on the webpage for SMIC, so feel free to have a look at that for a bit more information. In terms of what's next, there are numerous opportunities that we have to present this data. And of course, I'm very fortunate to be sharing it with you all today. We also have the winter poster event coming up at SHU, where I am planning to showcase a lot of these findings. And it is hoped that the data we have so far may be published in a relevant journal. Over the next few months, I will be focusing on building on the pilot data set I have so far, both for baseline testing and for low-level CO pilots. So it'll be really exciting to see those. Once pilot runs are completed, I will then be starting the MRI scan section of my project, which will sit alongside TCD data. We are also planning to obtain blood samples from participants pre and post CO inhalation to investigate what other biological mechanisms may be impacted by the presence of low-level CO within the body. I'd like to say several thank yous, particularly to my Director of Studies, Mari, who is at SHU, Caroline, who is also at SHU, and Paul, my third supervisor, who is at the University of Sheffield. An extra mention to the technical team at SHU, who have been particularly helpful with getting me set up with all of my pilot testing and my equipment. I'd like to say another huge thank you to the CO Research Trust and especially Adrian for inviting me to speak today. I'll leave you here with my references and of course welcome any questions that you might have for me.